Mesdames et messieurs, bonsoir. Ou rebonsoir à ceux qui étaient déjà là en début de semaine, lundi et mardi. Bienvenue à cette troisième conférence du Colloque Wright, dont c'est la 17e édition cette année. Cela fait effectivement déjà 30 ans que la Fondation Dudley Wright s'engage pour la diffusion des savoirs dans la société. Ça fait plaisir que, même un jeudi soir, de voir que la salle ne désemplit pas. C'était effectivement déjà bien rempli, complètement plein, même lundi et mardi également. Mon nom est Olivier Dessibourg, je suis responsable de la rubrique Sciences au journal Le Temps, qui est partenaire de l'événement, et c'est moi qui vais vous accompagner tout au long de la soirée. Comme chaque année, vous le savez, on a le privilège ici d'accueillir à Genève, à l'Université de Genève, des orateurs de renom qui viennent du monde entier, qui viennent partager leurs recherches, qui viennent réfléchir ensemble, interagir ensemble, discuter ensemble et surtout discuter avec vous. C'est une chance assez grande qu'on a d'avoir des orateurs de ce calibre-là venir discuter avec le public comme ce soir. Cette année fera évidemment pas exception. Vous le savez, le programme était riche. On a déjà eu en début de semaine même une prix Nobel, dans la personne de Linda Buck, qui est venue parler de ses recherches sur les gènes qui codent pour les récepteurs de l'odorat, pour lesquels elle a donc reçu le prix Nobel en 2004, de médecine et de physiologie. Tous les orateurs, cette semaine, parlent sur un thème, c'est la tradition. Ce thème, cette année, il est sous vos yeux. C'est décoder le livre de la vie, la révolution génomique. Pourquoi ce thème? Pourquoi est-ce qu'il a été choisi? Pour vous en dire un peu plus sur le choix de ce thème, je passe immédiatement la parole à Yvan Rodriguez, qui est professeur au département de génétique et évolution de l'Université de Genève et aussi membre du comité scientifique de la Fondation Wright.
Merci. Mesdames, Messieurs, chers collègues, je me réjouis, comme les jours précédents, de vous revoir ici pour cette troisième conférence de la semaine du Colloque Wright 2016. Colloque, comme Olivier Dessibourg vient de vous le mentionner, consacré à la révolution génomique. Pour ceux d'entre vous qui avaient assisté aux deux précédentes prestations de la semaine, eh bien, vous aurez certainement compris l'articulation de ces conférences qui, chacune, va pointer vers une direction différente, chaque fois aussi une direction liée à la génomique. La première conférence, comme on vient de vous le rappeler, celle de Linda Buck, était ancrée dans le présent. Dans le présent parce que les découvertes de Linda Buck nous font mieux comprendre les mécanismes de la vie, les mécanismes qui nous permettent à nous d'être ici aujourd'hui. Elle nous a montré comment l'identification de la plus grande famille de gènes codés par le génome des mammifères nous permet d'extraire de l'information du monde chimique qui nous entoure et de la transformer en percepte. La deuxième conférence, il y a 2 jours, qui nous a été présentée par Michael Snyder, celle-là nous fait nous diriger vers l'horizon, vers le futur, entre autres vers une médecine personnalisée, une médecine basée sur le génome, les génomes individuels. Aujourd'hui, la conférence de Svante Pääbo va nous faire, au contraire, nous retourner et nous intéresser au livre, au génome, qui nous parle de notre espèce, mais de l'histoire de notre espèce, de son évolution, et également de l'histoire des espèces, de nos espèces voisines espèces qui ont aujourd'hui disparu, mais espèces qui, en temps, non seulement ont partagé avec nous une partie de la planète, mais aussi, apparemment, partagé nos lits. Demain, nous allons, en écoutant le philosophe et essayiste Peter Sloterdijk, donner en temps à la réflexion, à laquelle nous pousse la compréhension, la réalisation de ce que représente la révolution génomique pour notre espèce. Voilà donc pour rappeler en quelques mots l'articulation de ce colloque qui dirige la lumière dans différentes directions, vers le passé, vers le présent et vers le futur.
Merci Yvan. Effectivement, ça, c'est pour les conférences, mais comme je l'ai dit tout à l'heure, il y a la partie interaction avec le public suite à chacune des conférences. On a voulu cette partie aussi interactive que possible, justement. Alors, à ce stade, je vous demande de ne pas éteindre vos portables, les mettre sur mode silencieux. Vous comprendrez pourquoi dans un instant. Il y aura les traditionnels micros dans la salle qui vous seront mis à disposition pour poser vos questions en live, donc directement. Et cette année, on a aussi voulu tester un autre mode d'interaction qui est Twitter. Vous avez ici l'adresse du Wi-Fi libre d'accès de l'Université de Genève, donc c'est Guest Unige. Et si vous avez un portable, et si vous l'avez mis sur mode silencieux, et si vous avez Twitter surtout, vous pouvez poser votre question à travers Twitter, simplement en mentionnant le hashtag, donc le dièse, suivi de Colloque Wright. Mentionnez-le bien, comme ça on voit votre question. Et Yvan va nous aider à les rassembler, à les trier et à les poser aussi directement. Donc ces deux modes d'interaction, pour permettre aussi notamment à ceux qui nous suivent en live, puisque cette conférence est retransmise sur Internet, de poser leurs questions à nouveau à travers Twitter. Cette session de questions-réponses se fera en français et en anglais, donc n'hésitez pas à poser vos questions en français. Vous avez dû recevoir un casque de traduction. Il y a une traduction simultanée. La question sera traduite simultanément en anglais aux personnes avec qui je discuterai. Je dis aux personnes parce qu'effectivement, il n'y aura pas seulement pour la discussion Svante Pabo sur la scène, mais aussi Peter Sloterdijk, l'idée étant justement l'interaction de toute la semaine, l'interaction entre les scientifiques qui sont présents. Voilà, pour vous parler maintenant de la conférence de ce soir, plus particulièrement et plutôt du conférencier, je repasse la parole à Yvan.
Alors, Svante Pabo est un biologiste suédois, qui s'intéresse à la génétique de l'évolution. Il s'intéresse à la paléogénétique, c'est-à-dire à la reconstruction des génomes d'espèces disparues ou alors d'organismes anciens. Pour ceux qui ne le connaissent pas par son nom, vous le connaissez très certainement par ses découvertes. C'est lui qui s'est intéressé au gène FOXP2. Alors l'acronyme est un peu rêche, mais c'est celui qui est communément appelé le gène du langage. Vous en avez certainement entendu parler, lu des articles dans les journaux. C'est Svante Pabo qui aussi a découvert une nouvelle espèce d'hominidés, en cousin, qu'on appelle l'homme de Denisova. C'est lui aussi, et encore, qui a séquencé une grande partie du génome de l'homme de Néandertal. Alors Svante Pabo commence sa carrière d'une façon un peu atypique, puisqu'il la débute comme traducteur dans les forces armées suédoises. Heureusement, il change de direction rapidement, si bien qu'aujourd'hui, il dirige le département de génétique à l'Institut Max Planck de Leipzig en Allemagne. Svante Pabo a reçu nombre de distinctions pour son travail de pionnier et ajoute à ses qualités scientifiques une qualité rare, en tout cas assez rarement associée à celle de scientifique, qui est celle d'être extrêmement clair dans ses présentations. Je cède maintenant la parole à Svante Pabo qui va nous faire voyager, grâce à nos génomes, à travers notre histoire. Dear Svante, welcome in Geneva. We're glad and honored to have you here. The floor is yours.
Well, thank you very, very much. Let me start by thanking the Wright Foundation and the organizers for the invitation to come to what is clearly an amazing week of science that you have here in Geneva. So what I wanted to start out by doing then is to remind you all about what you already all know. And that is the fact that our genome is made up of about 3 billion nucleotides, or letters, if you like, that make up the genetic information. And you also know that each of us carries 2 copies of a human genome, one from our mothers and one of our fathers. And these 2 genomes are similar to each other, but not totally identical. If you compare the sequence of letters and nucleotides, you will find a difference approximately every 1,200 or 1,400 nucleotides if you compare the 2 genomes that you carry in your body, or you compare 2 genomes between 2 people in the audience. And these differences then go back to mutations that have happened in the germline of your parents, your great-grandparents, and back through the generations. So each of us, if you compare 2 genomes, you have about 3 million differences. That is a record of the history, if you like, that separates those 2 genomes from each other. So our genome is an amazing resource to reconstruct human history. So if you now study the variation in the genome, and you start by doing that by simply looking at humans from different parts of the planet. So you look all over the world, what you will find is something rather surprising at first view. You will find that most variation is found among people in Africa. So although most people live outside Africa, of course, maybe there is something like 700 million people living in Africa and everyone else outside, the entire variation outside Africa is less. And that is due to the fact that most DNA sequences outside Africa have close relatives inside Africa, but there is a part of the variation in Africa that have no close relatives outside. And the interpretation of this pattern is then that modern humans originated in Africa, accumulated variation there, and then part of that variation, if you want, went out and colonized the rest of the world. So that is the genetic evidence, if you like, for the African origin of modern humans. And with some genetic tricks, you can also estimate approximately when one left Africa. And it's rather recently in evolutionary terms. It's less than 100,000 years ago or so. So this is then the recent African origin model for modern humans. There is also paleontological evidence to support that model. But it has a problem with it, if you like. If modern humans come out of Africa just 50, 100,000 years ago, there were other forms of humans there since a long time, since hundreds of thousands of years. There existed in Western Eurasia and Europe what we call Neanderthals. There existed other forms of humans in Asia that are less well understood. So these Neanderthals that then existed in this part of the world, here is a reconstructed skeleton of a Neanderthal compared with a present-day human. With these robust forms of humans, they appear in the fossil record somewhere 300,000–400,000 years ago in Europe and Western Asia. And they exist there until about 30,000 or 40,000 years ago when they disappear, in connection with that modern humans appear on the scene. And our laboratory in Leipzig are then fascinated by Neanderthals, sort of obsessed with them, if you like. And you may sort of wonder, why should we be interested in Neanderthals? And I think, to me, there is one really major reason for this, and that is that they are our closest evolutionary relatives, the closest relatives of all humans today, no matter where we live. So if we want to define ourselves as a biological group, or even species, if you like, it's really them we should compare ourselves to and say, what do we have all in common that they do not have, if we should define who we are? There are other reasons to be interested in Neanderthals too, of course. One is that they were here quite recently. Only 2,000–3,000 generations ago, there were Neanderthals here. So you may ask, what happened to them? Why did they disappear? Are they part of our ancestry? And so on. And this last question, I should be cautious when I speak. I'm sure there are paleontologists in the audience. But that question has been discussed in paleontology over decades. And there are sort of two extremes view of this, what happened to Neanderthals. One is that modern humans comes and totally replace Neanderthals, meaning there is 0% contribution from Neanderthals genetically to people today. At the other extreme, there would be a view saying Neanderthals are the direct ancestors of people in Europe today, 100% continuity. Hardly anyone believes that, but you can, of course, imagine anything in between here. So the first chance to test this with genetic means then we got in the early 90s, when we got a chance to sample not just any Neanderthal, but the Neanderthal, if you like. The Neanderthal specimen that was found in 1856 in Neanderthal in Western Germany and gave its name to this group of hominins. So we got to sample the upper arm there. At that time, we needed rather big pieces of bone to extract DNA. And we could then already then sort of rely on 20 years of experience in how you work with the tiny amounts of DNA that may be preserved in such bones. One thing that sort of is very scary is contamination from your own DNA. So for example, dust particles that you would have in a room like this when there are many people are often skin fragments, and a single such dust particle may contain a lot more DNA than in the bone we are studying there. So you have to dress up in funny clothes and filter the air and do lots of things to avoid contaminating your experiments with your own DNA. So at the time, we studied a tiny part of the genome, the mitochondrial genome that we inherit from our mothers. And you focused on that because there are many copies of this genome per cell, so it's easier to study. Looked at the particular variable part of that genome and found that if we sort of reconstructed the history of the Neanderthal mitochondrial genome, it went back quite far to a common ancestor that it shared with the mitochondrial genome of everyone living today. So whereas all humans today, their mitochondrial genomes go back to an ancestor 100, 200,000 years, you had to go back over 500,000 years ago or so to a common ancestor with the Neanderthal mitochondrial genome. And we and others then sequenced other mitochondrial genomes from Neanderthals. They all fall outside the variation of present-day people. So it is quite clear there is no person today on the planet that runs around with a mitochondrial genome of a Neanderthal. So in this scheme of things, it's total replacement, no contribution from Neanderthals. But it's also very clear that the mitochondrial genome is a very special part of our genome. As I said, there are many copies of it, hundreds of copies per cell, comes only from the mother, inherited as one unit, whereas the vast majority of our 3 billion base pairs or nucleotides are stored in the nucleus and inherited both from mothers and fathers. So the full story is in the nuclear genome. But there's, of course, only 2 copies of that genome there per cell, whereas there are hundreds of the mitochondrial genome. So I fear that about 10 years ago, I'm quoted in some newspaper article saying we will never sequence a nuclear genome from a Neanderthal. It's too little of it there. It's too degraded. It's chemically modified. It can't be done. And I guess that just goes to say that you should never make negative predictions like that, especially not in biology. Because what often happens is that you're overtaken by history, if you like, and generally by technology. And what came about in the early parts of this millennium were new techniques to sequence DNA, many, many fragments of DNA, cheaply and inexpensively and rapidly. So instead of now looking for a certain part of the genome you're interested in a bone like this, you could simply extract all the DNA in a bone, make a sort of library of DNA molecules, and just sequence all the DNA that's there. And start comparing it to the human genome, to bacterial genomes, and so on. So the first place where this worked for Neanderthals was in this site in Croatia, Vindija Cave. This bone, this fragment of the bone here, that is around 40,000 years old. And the first thing that you then notice, if you look at the DNA fragments you can find in such a bone, is that they're really very short. Whereas if you extract DNA from a blood sample from me, you will easily get 10,000, 20,000 base pair long fragments. Here, there are 40, 50 such letters or nucleotides, hardly anything longer than 200. So it's degraded to short, short pieces. It's also a very small fraction of the total DNA that comes from the Neanderthal. Our very best bones is 3 or 4%, much more typically below 1%. And the vast majority of the DNA comes from bacteria, and fungi, that have colonized the bone then over tens of thousands of years when they were buried in the cave. So we were very lucky to be funded over 5 years to improve the methods with which we come from the bones, extract the DNA, and convert it into form that we could feed into these sequencing machines. And we got a lot more efficient in doing that. We looked through many Neanderthal sites and many Neanderthal bones to find those that had the highest percentage endogenous DNA. We focused in the end on 3 bones from 3 different Neanderthal individuals from that site in Croatia, and sequenced a bit over 1 billion DNA molecules from those bones, which at the time was quite a lot. Today, you sort of do that easily. The vast majority of these DNA fragments were then not from the Neanderthal, but from bacteria. But we sort of then look at all these sequences and look which of them have similarity to the human genome, also taking into account that there are certain types of chemical damage that has accumulated in this DNA over time that give wrong nucleotides at different positions. And we sort of map this to the human genome then. And back at 2010 then, we had sequenced about 3 billion nucleotides from the Neanderthal genome, but in such random little fragments. So sometimes in the position in the human genome, we had 2 fragments covered, sometimes even 3, but we also had huge parts that we had not covered. So we had about a little over half the genome seen once or more times. But we could then begin to ask questions. And one of the first questions was, of course, this: what had happened when modern humans met Neanderthals? Did one mix with each other or not? So when modern humans come out of Africa and meet Neanderthals, what happened? So if there was a contribution from Neanderthals into modern humans, into Europeans today, one consequence of that would, of course, be that Europeans today should share more genetic variants with Neanderthals than people in Africa today. Since there have never been Neanderthals in Africa, there's no reason to assume that there would be any contribution from Neanderthals to Africans. So that's the first question we set out to ask. So it's this idea here: if this is a Neanderthal genome, if there is no connection here between them and present-day Europeans, this Neanderthal will be just as far from people in Africa as in Europe. If there is a contribution, then on average, the Neanderthal will be closer to Europeans than to Africans. So we had the Neanderthal genome from Croatia. We went out and sequenced 5 present-day people. At the time, there were not so many genomes known, and we wanted to have exactly the same error profiles so in these genomes. So we wanted 1 person from France, from Europe to represent Europeans. We debated a while and came to the conclusion that obviously a person from France is the most typical European. So we sequenced 1 French person. We sequenced 2 individuals from Africa, 1 from China, and 1 from Papua New Guinea. And then we did a very simple test to begin with. We just simply compared 2 present-day people, and to illustrate this and test it, we start with the 2 Africans. We just find all positions where they differ from each other, 2 present-day people. And then we ask, how often does the Neanderthal match what this present-day person has and this present-day person? Since Neanderthals haven't been in Africa and are not expected to have contributed to Africans, it should be 50-50. It shouldn't be closer to one African than the other. And indeed, that is the case. Statistically speaking, it matches as often this African as this other African. When we then looked at a European individual and an African individual, to my surprise, I should say, it actually turned out to be statistically significantly more matching to the European individual, suggesting, not proving yet, but suggesting that there could have been a contribution from Neanderthals to Europeans. Even more surprising to me was that when we did the same with a Chinese person and an African person, we again see more matching to the Chinese person, although most people would think there had never been Neanderthals in China. But maybe it has, who knows? But I think everyone would agree that there have never been Neanderthals in Papua New Guinea. And again, we see more matching to the Papua New Guinean. So everyone outside Africa we looked at has this more matching to the Neanderthals than to the African. So the question was, how could we explain this, that we see more matching not only in Europe where Neanderthals had lived, but in China, in Papua New Guinea, and Australia? And the model that we suggested, that have since largely proven right, is to say that when modern humans come out of Africa, they presumably passed somewhere through the Middle East. And we know there were Neanderthals there. So if these early modern humans, somewhere 50 to 100,000 years ago, were then those that became the ancestors of everyone outside Africa, and if they mixed with Neanderthals, they could, so to say, carry with them this Neanderthal contribution out into the rest of the world, also to areas where Neanderthals had not been. To the extent then that if your roots are outside Africa, somewhere between 1% and 2% of your genome comes from Neanderthals. So we were quite happy when we published this, that there were a lot of scientific follow-up very rapidly. People tried to find something wrong in what we had done, really couldn't find that, and started to follow up on the work, which is very satisfying. But I can never sort of stop myself from pointing out at this point that the general public also turned out to be very interested in what we had done. So we started very rapidly getting lots of mail, emails, and letters from people who self-identified as Neanderthals and volunteered to send us blood samples and hair samples that we should study. And after a while, I started seeing a pattern in this correspondence. It was almost exclusively men who said they were Neanderthals, and very few women who claimed they were Neanderthals. So I sort of counted all these emails and presented it to my research group as sort of my research. I count emails, and they were, of course, hypercritical of anything I say. So they said, this is just ascertainment, if you like. Men are more interested in genetics. They will write to you. Women are less interested. But I went back to my emails and found that that is not true, because there were plenty of women that wrote to us and said they were married to Neanderthals. And very few men who are married to Neanderthal women. So as a geneticist, you, of course, get very interested in an inheritance pattern like that. That's something you should follow up on. But we do other things too since then than counting emails. Particularly work together with archaeologists from Russia, and particularly at a site in southern Siberia on the border to Mongolia and China called Denisova Cave. So it's a beautiful place, if you ever had a chance to go there. And in 2010, they found a toe bone there, a phalanx of a toe, that turned out to come from a Neanderthal. And by that time, we had new super sensitive techniques to sequence small amounts of DNA, and it was well enough preserved. So we can go from this bad Neanderthal genome, where we had only a little over half of it from these different fragments here, to having a very good Neanderthal genome, where on average, every position is covered by 50 independent DNA fragments. So for the parts of the genome to which we can map these short fragments, this genome is actually of the similar quality or better quality than a genome we would sequence from a person today. So with this very good Neanderthal genome, we can now sort of go and look in different people today. So this is just one chromosome from different people. And in red is indicated the fragments that are similar or identical to Neanderthals and are inferred to have come from Neanderthals. So you will see that people generally carry different parts from the Neanderthals, but it adds up to something like 1% to 2% if you're from Europe or Asia. So you may then ask, is that a lot? If I carry 1% or 2% from a Neanderthal, is that a lot or very little? It's sort of a funny question. What's the answer to that? It is what it is. But maybe just to get the perspective on it, you might think about your own family tree. If you are here, you, of course, have 50% of your DNA from your mother and father, 25% on average from a grandparent, One more 3 generations back, 12%, 6%, 3%. And if you go back 6 generations, you have about 1.5% from an ancestor there. So if you like, it is as if one of your ancestors, 64 ancestors 6 generations back was a Neanderthal in terms of the amount of DNA. But the pattern in your genome is, of course, different. The Neanderthal DNA is in many small fragments. What you have from a person back there would be in big, big chunks. So another question you can now ask is, of course, how much of the Neanderthal genome is still there today and walks around on 2 legs, if you like? Because you can see we carry different parts. You can sort of add up sort of the Neanderthal genome, and that's indicated in blue here. And somewhere, it's a bit unclear, but in the order of 40% or so of the Neanderthal genome still exists there in people today. But our Russian colleagues find even more fascinating things at this site in the Altai Mountains. In 2008, they were very skilled, I think, to find a very tiny bone and not miss it, and recognize that this bone might be a human bone, and it's a part of a finger phalanx of a pinky of a child. So we sequenced the genome also from this bone, and we're very surprised. We got a good genome from it, and we're very surprised to find that it was not a modern human. It was not a Neanderthal. It was something else. It had a common ancestor with Neanderthals, but very far back. So if you compare this genome to the Neanderthal and present-day humans, say the deepest split between present-day human populations is somewhere in the order of 100,000 years or so. Then the split between this individual and Neanderthals is about 4 times deeper than that, about 400,000 years ago or so. So we sort of realized we had found a new group of extinct humans, and after a lot of thinking, we decided together with the Russian archaeologists to term them Denisovans. So just like Neanderthals are called Neanderthals after the site they were first found, these guys are called Denisovans after the Denisova site. If you like, it's the first group of extinct humans that are described exclusively from a genome sequence and no morphology or stone tools or anything like that. So have they mixed with present-day people? Indeed they have. And particularly, they've contributed to people in the Pacific, so Aboriginal Australians, people in Papua. So although the genome, the bone was found here, the traces of them in present-day genomes are particularly outside in the Pacific, and up to 5% or so of the genomes. There is also some contribution in mainland Asia, but it's mainly out here. So this, of course, suggests to us not that these Papuans have been in Siberia in the past, but probably that these Denisovans were more widespread in the past. So if I should just summarize what we think we know then about the origin of Neanderthals and Denisovans and modern humans from studying genomes, there is some origin in Africa for ancestors of Neanderthals and Denisovans. They come out somewhere perhaps 500,000 years ago, and in Western Eurasia then evolve into what we call Neanderthals, with the typical morphology paleontologists recognize. In the East, they evolve into Denisovans. We don't know how widespread they were. I've just sort of megalomaniac attempt to spread them all over here. We don't know where the border was between Neanderthals and Denisovans, but in this part then in the Altai Mountains, at some point there were Neanderthals, at some other point Denisovans. Then modern humans appear in Africa. Around 50,000 or a bit before, they start spreading seriously out across the world and mix with Neanderthals, presumably in the Middle East somewhere, and continue spreading out. There's rather good evidence now that there was subsequent mixing also in the ancestry of Asians, for example, because they have slightly more Neanderthal DNA than people in Europe. There is mixture with Denisovans somewhere in Southeast Asia. People continue out here. And then these other forms of humans disappear, but live on, if you like, today in this Neanderthal contribution and the contribution in Papuans. So Papuans would have 1 or 2% Neanderthal, and on top of that, 5% Denisovan DNA. So we've now found two cases, probably 3 of admixture. I wouldn't be surprised if in the future one found others, say in China. And also when modern humans spread across Africa, there is some evidence from present-day genomes that there has been mixing with now extinct forms also going on in Africa. So one message from this is that we have always mixed, not only among modern humans, but also with other forms of humans, at least to some extent. And we got a reminder of that recently from this site in Romania, a cave site where cavers found this mandible. It's a modern human mandible. It's around 40,000 years old. So it's among the earliest modern humans with skeletons indistinguishable from ours that we know from Europe. So when we study the genome from this mandible, and just look on one chromosome here, these are different other modern humans where we indicate with color pieces that come from Neanderthals. So this individual, we were very interested since it lived at a time when there were Neanderthals still around in Europe, if it had already mixed with Neanderthals or not. And the answer is that yes, it has, and it carries a lot more Neanderthal DNA than people today. And in fact, all these black parts are parts where we have no information. So over half of this chromosome is solidly of Neanderthal origin. So that, of course, means that this individual had a Neanderthal ancestor very recently back in its family history. And we can find 7 such regions in its genome where it has big chromosome parts from Neanderthals. So if we now look at the family tree of this person 40,000 years ago, we can infer that 6, 5, or 4 generations ago, this individual did indeed have a Neanderthal ancestor. So we almost caught them in the act, so to say, in a meeting. So we know then that there has been local mixing going on also here. So very clearly we have rejected this total replacement idea. There was a contribution from these other forms to present-day humans of a few percent, but we shouldn't lose track of that. The vast part of our genetic variation still comes out of Africa. To sort of not forget or lose that perspective, we like to call this model of modern human origin sort of leaky replacement. The big picture is one of replacement from Africa, but there is some contribution from these other forms. So the last few minutes here, I wanted to change the topic a little bit and say, well, what does that have for consequences today? Functional consequences. Are there contributions, genetic contributions from Neanderthals that actually mean something for people who live today? And that is something that we learn more about almost every month now. There appears papers illustrating, contributing to this. So what you can now do is go over hundreds of genomes in Europeans and plot how frequently Europeans have Neanderthal DNA at different parts of the genome. And you will see that it's a few percent, but here and there are things that very many Europeans have, 50, 60, 70%. Some of these are shared with Asians, some are unique to Asia, and some are unique to Europe. If you just ask the question, what types of genes are in those regions that have risen to high frequency that may have been advantageous for our ancestors that made them increase in frequency? The answer is rather boring, I think. It's keratins, it's sort of genes involved in skin and hair. So we don't know what these variants sort of have for consequences, but in the future we'll probably learn that some aspect of skin or hair morphology or physiology will come from Neanderthals. But there are also other things that pop up. So there was, 2 years ago, there was a gene variant found that is associated with type 2 diabetes, so the type of diabetes you get at old age. And this gene, the risk variant, encodes a lipid transporter that is quite different from the protective variant. This variant exists not in Africa, but outside Africa, especially in Asia and in Native Americans. And if you make a tree of the risk alleles and the protective variants and the risk variants, you find the Neanderthal right in here. So this is clearly a variant that have come over from Neanderthals into Asians and risen to high frequency. And you may ask, why should something rise and become frequent when it causes a problem, it causes a disease? And I think it's a good example of that genetic variants really interact with the environment to have their consequences. I think there is good reason to assume that genetic variants that today, when we eat almost too much through our whole life, cause us to have diabetes, those variants may actually be advantageous in a situation where you have starvation. So it may be that this is a sort of Neanderthal adaptation to starvation that we see. So have Denisovans also contributed something? Yes, they have. And the most dramatic example of that you find in Tibet, on the high plateau in Tibet, where people live at 3,000–4,000 meters altitude, where there's rather little oxygen in the air. People that move there from, say, Han Chinese that move there have quite a lot of complications, for example, in pregnancies. But it was already known that there were genetic adaptations in Tibetans to living at these high altitudes, particularly in a gene called EPAS1. And Rasmus Nielsen's group in Berkeley showed that this variant then exists very frequently in Tibetans, 80% or so. It's very rare at other places. And they showed that this variant actually comes over from Denisovans into the ancestors of Tibetans. So it's rather fascinating to think that we would probably not have so big human populations on the high plateau in Tibet if it wasn't for this contribution from Denisovans. So there are more and more such variants found almost every month, as I said. This is just another example from a research group in our institute. I wasn't involved in this. This is a variant in yellow here that has this suspicious pattern of being present outside Africa and hardly at all inside Africa, probably through back migration here. So this encodes, these variants are in genes that encode receptors in the immune system that has to do with how you defend against bacteria and viruses, sort of innate immunity. And you can actually show that if you're homozygote, if you have two Neanderthal versions here, you express more of these variants than if you're heterozygote and have one Neanderthal variant or no Neanderthal variant at all. And if you then look at what these variants are associated with statistically in the population, these Neanderthal variants are associated with increased resistance to Helicobacter pylori infections, so the bacteria that give you ulcers. So if you don't have ulcers, you may perhaps thank Neanderthals for that today. But there's another side to it. These same variants are also associated with increased allergic problems. So if you have allergies, you may perhaps blame Neanderthals for that. And just today appears a paper, actually, where people have looked across the whole genome for things that seem positively selected, gene variants in red here. And if you look overall on this, there's particularly genes involved in immunity that pop up here, and things that actually have to do with skin pigmentation and with other forms of eye color. So there's a picture emerging where these newcomers come out of Africa, the modern humans come out of Africa into Eurasia. They meet these other forms that have lived for 300,000, maybe 400,000 years there, adapted to the local environment. These newcomers from Africa mix a little bit with them, pick up advantageous alleles, and they sort of increase in frequency. So the sort of what one would call adaptive introgression if you studied animals or so. But I should also say that most Neanderthal alleles that are now pop up in the literature don't seem to have been positively selected. They are often associated with diseases because they come from medical genetic studies, and they involve several skin diseases actually, but also things such as depression, addiction to tobacco, and other things. But finally then, I think what is almost most interesting to me is now to study what we did not inherit from Neanderthals. So you can now go over the whole genome, all the chromosomes here, plot this here about how often we have things in Europe and in Asia from Neanderthals, and then ask, are there parts of the genome where we would statistically expect to see a contribution from Neanderthals if it just happened across the whole genome? But we don't. And there are such regions you can find. We would have expected Neanderthals to have contributed, but they have not. So it is as if we select against Neanderthals. We don't accept them in those parts of the genome. So we are so interested in them, these regions, because there is a sort of idea they could hide something that is indeed defining modern humans as a group. So things that changed very recently in our evolution here after we separated from Neanderthals. And why are we so interested in those things? Well, I think there are some things where we truly differ from Neanderthals as a group. If we think about Neanderthals, for example, they existed then for 3-4 times longer than modern humans have existed. But their stone tools at the beginning of their history, at the end of their history, experts will be able to say what's the difference between them. But they look very similar to me as a naive person. But I don't have to be an expert to say that modern human technology 100,000 years ago and today is very different. So something that comes with modern humans is technology that starts changing rapidly, also become regionalized because it changes so rapidly. So it will look different in Europe and in Central Asia very quickly, for example. Figurative art is something that comes only with modern humans, art that really depicts something. There are many papers appearing where people sort of want to say they find Neanderthal art. Sometimes they sort of say, well, but it's very modern art that they make, if you like. You can't see what it depicts. But things like this come only with modern humans. And of course, modern humans are the first group of hominins that are not anymore just say 100,000 individuals at a certain time. Their population size explodes to become millions, and you spread out of Africa, not just to Eurasia where other groups have been, but across water you come to America, Australia, and every habitable part of the planet. So this really seems to have something to do with culture and how you interact with each other, perhaps. So now when we have the Neanderthal genome, we can make a catalogue of all features in the genome, all changes in the genome, that everyone has in common today, no matter where we live on the planet, but the Neanderthals look like the apes. The things that changed here, and we all share them. And the fascinating thing to me is that that's not a long list. So whereas 2 genomes today, as we said in the beginning, differ at 3 million positions, if we now make the requirements we should all share something and the Neanderthal should look like the apes, it's only around 30,000 differences. So you can look through them in the computer in an afternoon. The problem is only you don't understand at all what you look at. The sort of dirty little secret of genomics is that we are extremely bad in inferring any function from just a change we look at in the genome. So, of course, we have a particular interest since we sort of think about that this has something to do with cognition, to sort of think about things in the brain. So if we just, to give you a feel for this, say, among these 30,000 changes, there are only 96 changes that change an amino acid in a protein, in the molecules that really do things in our body. So you can look at all these proteins, and then you can sort of say, does any one of them look interesting? And then we are particularly interested in genes and proteins active during development of the brain or function of the brain. But that takes us then to the very last question I wanted to bring up, and that is, how should we then study such changes when we have candidate changes that we think are important? Well, I sort of go around since many years and make jokes in talks like this and say, what we want to do with these Neanderthal-like variants is to put them in a transgenic human and we want to take the human variants and put them in a transgenic chimpanzee, and then we want to study their cognition. And we can't do that because there are ethics committees and lots of things that we have to deal with. But it's not just a joke, because there are now even professors at Harvard who sort of start going around and saying that now when we have the Neanderthal genome, we should edit the genomes of a stem cell and have an individual bone and to sort of clone a Neanderthal. And I think it's sort of quite ridiculous, both for technical and ethical reasons. We will not do that either. But what can we do about these things? Well, I think one thing we can hope to do in the future is to look for back mutations in the human population. So every baby that's born has something like, say, 50 new, maybe 100 new mutations. There are 7 billion people on the planet. The genome is just 3 billion base pairs. So every mutation compatible with human life exists out there in the population, but at very low frequencies. But I think in the future, when we all get our genome sequenced as soon as we go to the doctor, I think we will start finding mutations back to the ancestral state. And I think there will be ethical ways to then study such individuals. What we can also do is engineer in these changes in stem cells and study cells in tissue culture, and we and others are doing that. And what I want to sort of illustrate a little to you is that you can also use, in some cases, laboratory models. And that brings us then to this gene that you heard mentioned in the introduction, FOXP2. It's a gene here that's particularly interesting to us since several years now, because Tony Monaco in Oxford identified it in a large 3-generation family where individuals are affected by a very specific language problem. There is no indication they have problems with intelligence or anything else. It's specifically sort of language production. And this family allowed them to identify the gene. It's a gene that encodes a transcription factor, so a protein whose function it is to turn on and turn off other genes. And the encoded protein, interestingly, have 2 changes in it. Although it's very conserved in general, it has 2 changes in human that are not there in other primates, for example. So we were very interesting when we started getting the first Neanderthal and Denisovan genomes, what they would look at these 2 positions here. And it turned out the Neanderthal and the Denisovan actually turns out to look as present-day humans. So these 2 changes then happen before the divergence to the Neanderthal here. But that doesn't necessarily make them uninteresting, and you can sort of illustrate how we will in the future study such human-specific changes maybe. So since this protein is very conserved between mouse and human, it has these 2 changes here only, and 1 very conservative change there that we hope does nothing. What you can do is to humanize a mouse. You engineer in these 2 changes into the mouse gene. And you then have a mouse that from its own gene makes a human protein, so to say. And since we think that this protein has something to do with speech and language, you then try to speak to the mouse, and it will not answer. So perhaps its lifespan is too short to really learn language or so. But you at least have some kind of an animal model that you can now study. So you can look in the brain of the mouse if it has any consequences. And interestingly, these humanized mice, if you compare them to their wild-type litter mates, born by the same mothers but not having these changes, they make longer connections, longer dendritic trees, so more long-range connections to other neurons. So you can see that here. And particularly in parts of the brain, the corticobasal ganglion circuits that have to do with motor learning. So that then caused a previous graduate student from us to take these mice when she had finished and go and do a postdoc with Anne Graybill, who's an expert in sort of these types of circuits at Harvard. So then they tested learning in these mice. So for example, they ran in a maze here, and they have to learn to go, say, to the left to find chocolate milk that the mice like very much. And you can give the mouse a cue, so to turn on a light, for example, and it knows it should go towards a light. It learns that. And there's no difference between the humanized mice and wild-type mice in how quickly they learn that. But it then comes, if you give always a signal to the left here, it comes a time when you can do away with the signal. And the mouse has simply internalized, I always go left, and I find my chocolate milk. And that switch, the humanized mice make in 7 to 8 days of training, whereas the wild-type sisters and brothers of them take 11, 12 days to learn. So they sort of automate muscle or motor activity quicker. So I'm not a neurobiologist, but the neurobiologists sort of tell me that this is, think about when you learn to bike as a kid. In the beginning, you think about what you do, and you're very bad at it. You sort of tip towards this side, and you just steer there, and it's all very counterintuitive. But then comes a point when you suddenly stop thinking about what you do. And then you're suddenly very good at biking. You have sort of automated this muscle coordination movement that you need to do on the bicycle. And that actually goes together with a switch of activity in a part of your brain, in a striatum, from the medial part here to the lateral part. That was known. So we went back and looked in our mice, and indeed we see this sort of activity switch more pronounced in the lateral part than in the medial part of the striatum, fitting with this idea. So the idea is now that what happens in this mice when you change these two positions is that you change these circuits here and make for faster automation of motor learning. And of course, if you think about language and speech, that is, I would say, clearly the most sophisticated muscle coordination thing we do in our lives. We have to have millisecond control between vocal cords, tongues, lips to produce articulate speech, and we take quite a lot of time to learn that as a kid, and there's quite a lot of variation in how fast we learn this too. So the idea is that these changes will then have something to do with sort of articulation particularly. Very last thing, this then happened before the split from Neanderthals. Have anything happened only in humans in this gene? Indeed it has. There is a region here with a change in this gene that has changed and is present in almost all humans today, but every other animal we looked at has something else. And this position has something to do with how the activity of this gene in the brain. So we can show that activity changed when we change this position here. So the idea now is there are these two changes in a protein here that we show in the mouse are important, and we think that some regulatory change here is also important that happens later. And this may sort of be a sort of a little illustration of what we might find in future, that there are several changes that happen on the way here to come to the sort of human condition. So I think this is sort of encouraging that one might be able to model aspects of even human-specific things, such as, say, articulation and speech in an animal model. So it's encouraging for other genes that have changed in recent human evolution and that may be involved in such things. So I hope I convinced you then, during this 50 minutes, that if you're interested in human evolution, and particularly recent human evolution, it's very good to have the Neanderthal genome. You can time things if they happened here or here. You can focus on these things if you're particularly interested in them. And what's really the challenge for the next 5 years or 10 years is to now go on and study the function of these changes that happened here. To do that, we will study sort of stem cells in vitro, but what one will also do, I think, is to make humanized mice to sort of study the effects of these things. So many, many people are involved in this, many more than I can mention. I want to mention one person, Matthias Meyer, who developed this ultra-sensitive method for sequencing DNA from bones that allowed us to get these high-quality genomes that we would not have had without his technical developments. Many people have helped analyzing the genomes. I sort of particularly mention Janet Kelso, who's head of bioinformatics in our institute, and Kai Prufer, who coordinated the Neanderthal high-quality genome effort. And for population genetics, particularly David Reich in Harvard, and the Broad Institute, and Monte Slatkin at Berkeley. And with that, I then thank you for your attention.
Thank you. Thank you, Svante Pääbo. Fascinating presentation. Please take a seat.
As a seat, okay.
Yes, so as I told you at the beginning, the idea of this colloquium is really to bring scientists together to discuss about ideas, exchange ideas about the issue of this week, which is the genomics. And tonight we have the chance to have a second speaker for the panel discussion. And that is Peter Sloterdijk. You might know him. He's a philosopher, famous philosopher at the Hochschule für Gestaltung in Karlsruhe in Germany. He wrote quite a lot of books, also on genomics. That's why he was invited to this colloquium as well. May I invite him to come on stage with me? You have to use this mic. Yes, please take a seat. And let me remind you that you can also ask your question through Twitter again. So it's very easy. Just go on Twitter, type the hashtag Colloque Wright. And I'll start with some questions maybe. I'm really curious, what did those people who sent you emails saying, I'm in Neanderthal. Oh, actually, what did they say in their emails? Did they say, I'm a Neanderthal, I have a parent that is Neanderthal? And what did they write, to describe that?
Yeah, there are, of course, a lot of different things, but some people, of course, generally it is people who sort of look or behave like we would imagine in popular culture in Neanderthal would. Some people send pictures, and, some of them are more that you would recommend them to go to an endocrinologist, acromegaly, when you have too much growth hormone or something like that.
And what does that mean to you, that some people compare themselves to Neanderthals today? How do you interpret that? And that's also a question for you, maybe.
Yeah, I think that people are fascinated by the idea that there were some other form of humans here rather recently that were sort of probably in some respects rather different from us after all. I think it's sort of fascinating to think about things like what would have been if Neanderthals would have hanged on for another 3,000 generations. It's not that much. If they were still here today, I sometimes ask, would they have lived in suburbia or would they have been in a zoo? And we don't know, right? We don't know what we would have done with them.
Fascination? Do you think it's fascination?
Yeah, fascination is absolutely obvious. And fascination always has its origins in the question, how? So we all know this famous term coined by the evolutionists of the late 18th and early 19th centuries, the term of the missing link. In that case, I would like to ask, there's not only a link that is missing, there's a missing encounter. I do not see how humans and Neanderthals could ever produce what you called interbreeding. Where's the meeting point? Where did the romance happen?
You said it was a surprise.
It is. It leads almost on a terrain that could also be that of science fiction, because the next question will automatically be, what about the genes of extraterrestrial invaders. If we could mix with Neanderthals, why not with inhabitants of the outer space? But one thing is certain: our ancestors cannot have been racists.
I don't know. But I always tend to say what we now speculate about what happened when we modern humans met Neanderthals and how these things happened. It really says something about our worldview. It says nothing about what happened back there. It says something about how we view humans. It depends on if you think romance is absolutely necessary to have sex. I don't know. Why are Neanderthals not there? You can say, if you're a pessimist, now I disappear.
Should be okay.
Hello? Yes. If you're a pessimist, you can say, well, obviously it was the first big genocide. Just see how we behave today, right? And they are gone, and we must have just killed them. You can turn that around and say, let's look at the Middle East. The earliest modern humans outside Africa we find in the Middle East at 120,000 years ago. The last Neanderthals in the Middle East are around 60,000 years ago. So there is 60,000 years of peaceful coexistence, and that in the Middle East. If we could have that today, I would be really happy. Let's start fighting 60,000 years again. And of course, we don't know what happened. It may be that modern humans were there and disappeared again or something like that. But it says something about how we see humans, not about what happened, really.
Well, you said it was a surprise that you found like those 1 or 2% of Neanderthal genes in our genome. But that was what you were looking for, right?
Yes, it was one of the things we were looking for. I think I was very biased. I've sort of written a book about this where I sort of describe my biases. Yes. That after seeing the mitochondrial genome, and that there was no contribution there, of course, intellectually we know we could very well, and we have said publicly, it could very well still be a contribution. But we were really biased to listening particularly to those paleontologists that said, no, we see no evidence of it. So I was surprised.
You mentioned that you didn't elaborate on the fact that Neanderthal disappeared. Do we know why? Do you have hints for that?
No. I think the only reasonable thing, if it was only Neanderthals that disappeared and not other forms of humans, I could say they were susceptible to some disease or something special. But, the Denisovans are gone. We know there were other forms, these hobbits in Indonesia, these Homo floresiensis, they are gone. In reality, the last 30,000 years or so of human history is unique in that we are alone on the planet. Up to that point, there were always other forms of hominins around. So it really must have something to do, I think, with our behavior. But what exactly that is not clear. It could have been different things in different regions of the world, too.
You described in many details what we inherited from Neanderthals. But if I take it more generally, what have we learned about us when we look at him now? That's also a question for you, maybe.
Well, I tend to be very concrete, since after all what we do is very concrete. Yes, we know the Neanderthals they are part of modern humans today still. About half of their genome is still around today. We know that we interacted with them, had babies together, and those babies must have been incorporated in the modern human society because they were successful and went on and had children, and otherwise we wouldn't see this contribution today. So we've learned something about our sort of ancestry.
From what you've heard, how would you answer that question? What have we learned about us when we look at him?
Yeah, my feeling is that the modern civilization as such, with its decision to replace myth of origins by evolutionary theories is running into a very deep predicament today, because we discover that there are that many empty spaces or black holes in our big narrative of evolution. That this big operation becomes more and more precarious. It was from the point of view of the narrative economy much more profitable to tell a story like Genesis. But now, but this story makes us strangers to ourselves, because the human beings usually cannot look into the mirror of genomics, because it's not a mirror that shows yourself your own picture. You see that you do not know yourself, and that you can make this enormous detour. But I had a feeling during your very fascinating declarations that so the story of Genesis and Jurassic Park are getting closer to each other, and that this kind of analyzing the human past is already several steps closer to the Jurassic Park logic than to the biblical way of looking at the things.
A last question before I turn to you, to the audience. If you met a Neanderthal in this room or outside, what would you ask him?
Well, I would first ask him just anything to see how the articulation sort of worked.
Okay, so Fox P2. Yeah.
And yes, I think a fascinating question would be, say, how do we differ cognitively? Is it sort of, I could well imagine that the Neanderthal individual is just as intelligent as I am, but that there is something about sociality that is, many people have argued that there's something very special about our sociality, that we're so interested in giving lectures and influencing people and spreading our ideas, and that may be what sets humans on this path to these big societies and a cumulative culture where we spend, say, the first third of our lives learning what previous generations have done and then add to it.
Could that have lost Neanderthal as well? That could be a reason for his loss.
Yes.
Okay, I'll now turn to you. Do you have questions for our two guests tonight? I'm sure there are some questions. There's one up there. Yes, please.
Ça marche? Ouais. Good evening. Thank you for this very interesting talk. I was wondering, you mentioned the discovery of a Homo sapiens that was 40,000 years old, who happened to have a large amount of Neanderthal DNA. And I was wondering, suppose we didn't know about the existence of Neanderthals, would you have been able to infer the existence of such things on the basis of the DNA? And then the second question is, could we imagine finding similar Homo sapiens in other parts of the world and inferring other types of Neanderthal-like ancestors, Devonian you mentioned, maybe others, based on... You understand my question, right?
Yes. Can we, from sort of studying present-day genomes, infer the existence of a contribution from some extinct form? I think in the case of that Romanian individual that had so much there, it would have stared us in the face. We would have realized that. There are people who have developed method a group in Seattle particularly, have developed methods where they sort of claim to be able to use present-day data to infer pieces that come from extinct forms of humans. I would say that without having the Neanderthal and Denisovan genome, one would still not totally have believed it. So I think it's incredibly valuable to have those genomes. In Africa, there are several groups of sort of found regions in the genome that have deep, deep divergences, that go back 1.5 million or so. And it's quite reasonable to say they come from some extinct group of humans. But before one has those genomes, it's very hard to fully understand the history. I could imagine, for example, that in Africa there's been a big meta-population over hundreds of thousands of years where one continuously exchanged genes with different groups. And actually modern humans is more appear in several places, if you like, for the things to come together. So I think it's still very valuable to have these genomes.
I'll turn to Twitter. Do we have Twitter questions?
Sure, we have quite a bit. There's one which is quite funny, I think. Do you think that the absence of Neanderthal mitochondrial DNA in humans suggests that Neanderthal women were not sexy?
Yes.
Because of course, mitochondria.
Yes. So if it was only Neanderthal men that contributed, mitochondrial DNA would not come over. You would expect other things too. You would expect less contribution on the X chromosome, because a man, of course, gives his X chromosome only to the daughters and not to the men. So you would expect half if there was only men, we would expect less contribution on the X chromosome. And we see that. But, so when we first saw that, I was sort of convinced. I said, yes, it's really modern human women that find Neanderthal men sexy. And that's reflected in the emails we get even today, that they marry Neanderthal-looking men. But then we found these other regions on the other chromosomes where we seem to have selection against Neanderthal contributions. So if you have that on the X chromosome too, it might be, that type of selection instead. So, if I have, yes, it's really the Neanderthal men that are sexy and not the Neanderthal women.
Maybe another one? Yes?
Yes, well, so this is very striking. In your drawings, we see that Neanderthal lived for hundreds of thousands of years in Eurasia and never crossed the sea. So what do you think? They were afraid of water? Because we did that very quickly.
Yeah. Of course, chimpanzees and apes don't swim at all. They sink as stones. That's why you can, in the zoo, just have a water moat, right, to keep them in. A human would swim over right away. We don't know how they were with swimming. I think it probably also had to do with technology to some extent. But I do think that if you look at hominins exist in Africa and in Eurasia for 2 million years, if we go back, they never come to Australia, never to America. They never come to Madagascar from Africa over all this time. So there is something there, I think. They spread generally just as other mammals. When you see land on the other side, you cross. Modern humans in only 50,000 years spread not only to these big continents, to every little island in the Pacific, right? How many people must not sail out on the Pacific and disappear before you found Easter Island? So that's why I often say that we are mad and they were not mad, right? You are mad if you sail out on the Pacific and not knowing if there is anything there. So I think, my bias is to think there's also something there in attitude to the world, and we never stop. Then we go to the moon, then we go to Mars,
There was one question just here behind. Is that correct? No? Pas anymore. So, Thierry, maybe?
Isn't it surprising that you don't find any Neanderthal DNA in Africa? Because it means that none of those who have faltered with the Neanderthals came back.
Mm-hmm. Mm-hmm. Yes, it's in general a little sort of like Professor Escoffier might have more opinions on this than I, that you see so relatively little back migration into Africa by modern humans. You can detect some, but that it's generally a movement out of Africa and out to the rest of the world. But yes. And the Neanderthal contribution you see in Africa is generally well correlated with a European component, that there is sort of back migration that brings it back. But Neanderthal certainly did not go back.
There's one further question here.
So I'm thinking about skulls. I think I've seen a Neanderthal skull somewhere with marks on it. And that might have been in Croatia, in that museum.
Can you speak louder?
I'm sorry. So I'm thinking about marks that are sometimes seen on Neanderthal skulls, like a bunch of vertical, horizontal marks. And sometimes the interpretation that I read, I remember, was perhaps it's some kind of art that they did on dead people or cannibalism. Do you have any opinion on that subject, perhaps?
I think there is quite good evidence. It's particularly a paleontologist called Tim White at Berkeley who has argued that he sees lots of evidence of cannibalism in Neanderthals, and even more than in early modern humans. If you see those, I don't know about this sort of marks on the crania, you say, I'm not aware of that, but there's lots of cut marks on the bones where muscle attachments are, for example. And if you look at those pictures of the bones I showed that we have used, they are sort of fragments like this, and there are particular bones with bone marrow. So it seems very like if one has crushed the bones to get to the bone marrow. And it's sort of very suspicious that, for example, the tibia here where there's bone marrow is crushed, but the fibula, the other bone here with no bone marrow, are generally intact. So what Tim argues is that they have treated this pretty much like any other, animal that they have. Then one can, of course, argue, have one eaten it for some kind of religious sort of purpose or not? But, and we even have the feeling that our best bones are the ones that were eaten. That there are Neanderthal burials where the whole skeleton has been put in the ground. There we generally find proportionally much less DNA because there are, of course, months of bacterial activity when a body lies in the ground. Whereas if you take a piece of a bone and gnaw off all the meat and throw it in a corner in the cave, it dries out pretty quickly without bacterial activity. So we actually thank the Neanderthals for the cannibalism, for sort of allowing us to get the genomes.
Well, when we're talking about the image we have of Neanderthal, I mean, like decades ago, like 2 decades ago, it was totally different than the image we have today. We had this image of a kind of brutal, stupid, like boorish man, like a bit, animal-like. And this image changed radically in the recent years. That's maybe also a question for you. Why? Why did it change? Why do we have such a renewed image on Neanderthal? Is that, the progresses in science and genetics? Is that something to do with religion? Why?
I would also say both images are still there in people. There's both this image of them being brutish, stupid, and violent, and this image of them being particularly sensitive and wonderful and being sort of exterminated by these mean modern humans that come. I think both things are sort of there.
Maybe new discoveries, because there was a discovery where they found a Neanderthal with flowers, I think, of presents in the tombs. So first they were buried, and they had presents with them in the tombs.
Yes, so there is one site like that where it has been claimed, this has been a bit disputed, is Shanidar in Iraq, where one sort of seems to be, yes. But there are clearly burials. If the flowers were there, I think is debated. But yes, and they sort of had, of course, technology at the time, say 50,000 years ago, that were not that different from modern humans. There's also evidence that late Neanderthals that could have met modern humans actually picked up some technology from modern humans.
Do you have a view on that? Why we changed this image we have on Neanderthal?
I would like to hear scientific commentary or a very naive question. Why do modern humans have the feeling that they are aesthetically more developed than the Neanderthals? There seems to be a kind of bioaesthetic drift in the evolution towards human beings. Yeah? So in general, I would agree with that. I would say that the human race is becoming more prettier and prettier. Maybe the Neanderthal did not really participate in that.
You mean
evolution among themselves? They were, as it seems, pretty enough to
So you mean the individuals becoming prettier, or about art, producing art?
Yeah, but no, I think that a certain kind of beauty is sort of congenital with the human race as such. Yeah, for instance, the
but
fact
the beauty is in the eye of the beholder, isn't it?
Yeah, we do not know the opinion of the animals, but human beings amongst themselves. Certainly have the feeling that a human face has something to say, and there's always a kind of beauty to it. And without that beauty, humans could not be what they are. And if we have drawn these maybe wrong consequences with regard to the Neanderthals, that they were too brutish. They had no metaphysical talent. They had no feeling for transcendence or something else. Everything the human beings are proud with regard to themselves. Yeah? If that has been missing, that could be an explanation why we did not really recognize them as members of the family. But for me personally, I would like to know if there is any hint from the point of view of this paleoanthropology that helps to understand that feelings that humans live under very particular bioaesthetic conditions.
You have an answer?
No.
Right.
No.
Question. Are there any questions?
And this is this difference between male and female. And the audience had short moments of generalized hilarity when it came to the question who married partners of the other side. There is a famous story about a Vienna cook, a female, who said, alles was ein Mann schöner ist als ein Aff ist ein Luxus. I wonder if you understand that every time a man is more beautiful than an ape, is luxury. And this is part of this everyday bioaesthetics that seem to be part of the game already in very, very archaic dimensions.
We'll turn to the. Are there any further questions? I can't see very well. Yes, there's one here.
Something more specific. Just for clarity, the previous speaker spoke of the origin of Neanderthal as coming from Africa, and that's clarity because I don't quite understand, and that they found a very small percentage of DNA in the African population of Neanderthal, and yet you did say that it originated the Neanderthal in Africa. You explained it that the reason why there's lesser DNA is because the Neanderthal never returned to Africa. Could you clarify that? I don't quite understand.
I mean, I think there is an origin for the ancestors of Neanderthals in Africa. Most paleontologists would call that Homo heidelbergensis, which is something that then, 400,000 years ago and further back exists both in Africa and in Europe. And this sort of contribution from Neanderthals that we see that are big parts of 30,000 or more nucleotides pieces that we find only outside Africa. Whenever we find it inside Africa, it's really correlated with gene flow from Europe into Africa that we of course have. People have moved sort of back to Africa over history.
And what is the significance? I mean,
the significance of the Neanderthal contribution?
Yeah.
Yeah, I mean, we begin to learn that some of the genetic variants from Neanderthals have medical consequences, for example, as I discussed towards the end.
But the origin is Africa.
Yes.
That has not been
origin
disputed.
of all hominins eventually.
One more question. Homo sapiens, I know it's hard to reply. When did the genus Homo become sapiens?
Well, I mean, so if you were modern, Homo sapiens mean modern humans?
The modern human. When it became the man of wisdom or whatever.
Yeah,
what clenched it?
Yeah, I mean, that's a big billion-dollar question for the next 5 or 10 years. I would hope it's in this catalog of changes we have found, and that's what why I want to study them.
One more question.
Oh, sorry, we will go to other persons as well, please.
Sure.
Twitter?
Yes, so this will rejoin one of the questions which were asked at the very beginning. How much of today's diversity of humans could result from breeding with still unknown Neanderthal-like species? And there are questions about the man of Flores.
Of the
Flores.
Flores, yes. Yeah. So I would be very surprised if I would not be surprised if one find other groups of now extinct hominins, for example in China, who have also contributed. I would be very surprised if that ever adds up to even approaching, say, 10% or something like that. Because we see a clear pattern that the variation come out of Africa and for the vast majority. So I would not be surprised if one finds other things, but they will still be small. It will still be a small proportion of the variation. So yes, there are these sort of short-statured hobbits like Homo floresiensis in Indonesia. There is no indication sort of that such groups had contributed to the gene pool. There is indication that something like that have contributed to the Denisovan genome. We actually see an old component in a Denisovan genome that's not there in the Neanderthal genomes, that comes from something that diverged 1 million years ago or more back in history. So that could be something like Homo erectus in Asia or something like that that contributed a few percent to Denisovans. So these other groups have also mixed with each other. There's a contribution from Neanderthals into Denisovans also, actually. So everyone has sort of mixed with each other a little bit.
There's a question in the back there. Yes?
Yes, thank you for your speech. I have a question about the longevity and the aging process of the Neanderthal, and because the former scripture sometimes describe humans who have lived for over 200-300 years, which we still don't know if it's a myth. Could you tell us more about the maximum longevity you have observed in the species you have studied?
Well, so I'm not an expert on this at all, but I do understand it's very hard to judge the age at death of skeletons after, 15 years of age or so, where the bones and teeth are in the adult state. There are people who have claimed that Neanderthals would have lived 4 or 5 years shorter on average than modern humans from the time. That could, of course, have made a big demographic influence. If you live in a society where you're sort of essentially pregnant continuously, it would mean one less child per generation. That could have a big impact. But yes, we don't know about maximum lifespan at all for these things.
I have a question on the experiments you did on mice, trying to put this Neanderthal gene sequences into mice. Is that possible? Can we imagine to do this on monkeys? Because monkeys are obviously closer to primates than to humans than mice.
Yeah.
And this is for you. And what do you think? What is your view on such experiments? Maybe first.
Yes, of course, monkeys, meaning not apes, but monkeys.
Non-human primates.
Okay, so non-primate would include apes, would be chimpanzees and orangutans also. And I think with apes, it's sort of totally excluded from an ethical perspective. Of course, one can imagine many things, but in the civilized world, I would say that's sort of what we have learned over the last decades about chimpanzees and gorillas and orangutans is that they are much more human-like than we had thought in their cognition, in their behavior. Of course, monkeys, meaning sort of primates with tails, if you like, are used as experimental animals still. I mean, it's debated if that is appropriate or not, but macaques or baboons are used. In China, for example, there is quite a lot of work on making transgenic macaques. Things like that will happen, whatever one thinks about them. I think it will happen.
With Neanderthal genes?
I think, of course, most of that work is focused on medically relevant things, but I think there will also be work in this area, yes.
What is your view on that?
I would like to ask a question if we are able to distinguish something like tribes or ethnic groups or cultures on the level of the Neanderthal?
Yes, I do think that paleontologists sort of see different, especially in late Neanderthals, where one have more, much more material. There are sort of cultures, in France, there is a thing called Châtelperronian, which are sort of late Neanderthals that seem to have picked up some things from modern humans. And
is there any evidence for warfaring between Neanderthal groups?
Of course, with war, you then mean some organized violence in some sport, not individual violence. And I don't think there's any evidence to sort of support it. But, you know, chimpanzees have warfare, if you like. There are neighboring groups that systematically take out the individuals from the other groups. So.
We'll take two more questions, one from the audience and one from Twitter.
Une petite question. Simplement, je trouve vraiment miraculeux qu'on ait pu retirer de l'ADN des petits fragments pour notamment Denisova ou autres. Ma question est, jusqu'à quand dans le temps en arrière peut-on espérer pouvoir encore trouver de l'ADN utilisable?
How far back can we go? Finding DNA.
So how far back can one go to retrieve DNA?
Expect to go or hope to go?
Yes. So the oldest DNA that is believable that has been retrieved today is from the permafrost. So it's been continuously frozen. And that's work from a group in Copenhagen that studied a horse genome. That's about 700,000 years old. The oldest hominin DNA is something that we have studied from a site called Cima de Luesos in Spain. That's 430,000 years old, or maybe 450,000 years old. It's very little DNA there, but we can sort of get a little idea about how this is related to other groups. And it turns out to be very early on the Neanderthal lineage. It's some Neanderthal ancestor, or pre-Neanderthal, if you like. But to say in the order, when it's very well preserved outside the permafrost, in the order of 500,000 years, or something like that.
Last question from Twitter.
Yeah, so are there any hints at what could have led to the demise of Denisovians and humans?
In other words, does their disappearance directly correlate with the arrival of Homo sapiens?
Yeah, so that would be better with paleontologists asking that question, but I understand many paleontologists at least say that when modern humans arrive in an area, Neanderthals disappear, and it seems rather quickly. But there are exceptions to that. I mean, Middle East is such an exception. There are early modern humans there 120,000 years ago. There are Neanderthals 60,000 years later still. It's then a question, were they really overlapping? They must have been in contact in some sort of zone of contact. But is it that modern humans are there when the climate is more African-like, when it gets cooler, the Neanderthals are there? That sort of, the fossil record is not complete enough to really say if one overlapped, actually.
I will ask the very last question. What is your dream? What, in the last years of your scientific life, do you dream to find or to discover?
I mean, if one could find one or a few of these crucial changes that set modern humans apart cognitively from Neanderthals, something like FOXP2, but in modern humans relative to Neanderthals, that's enough of a dream for me. Then I'll die happy.
A good dream. Thank you very much, Sanjay Pablo. Thank you, Peter Sloterdijk. Voilà, il me reste à faire 2-3 remerciements à la Fondation Wright qui organise ces colloques, à l'Université de Genève qui les accueille ici, à vous d'être venus, aux organisateurs, aux traducteurs en direct, on les oublie des fois, merci à eux. Et à vous donner rendez-vous pour la dernière conférence de Peter Sloterdijk, demain soir, même heure, même place. Si vous avez aimé cette conférence, dites-le nous. On organise un petit sondage pour améliorer chaque année ou chaque 2 ans ces colloques. L'adresse est la suivante, colloque.ch/sondage. Dernière chose, vous savez qu'il y a un spectacle qui est lié à ce Colloque Wright tous les soirs à 3 heures différentes. Je ne les ai plus en tête, mais vous trouvez ça sur le site. Jusqu'au 20 novembre au Parc des Bastions. Allez le voir, il paraît que c'est fantastique. Merci à tous. À demain pour ceux qui souhaitent revenir. Bonne soirée.
Thank you.
Thank you very much.
Yes, it was fun.